day. Could you please introduce yourself and tell me what course you teach?
My name is Bill Brieher, and I teach MCB 471, which is Advanced Cell Biology, Cell Structure, and Dynamics.
When is your course typically offered?
We offer the class in the fall semester.
Can you please give me a brief overview of what is covered in your course and what are the main learning goals?
We cover the structure and dynamics of the typical eukaryotic cell. Sometimes we investigate prokaryotic cells because that can help inform us as to how eukaryotic cells are organized as well. The goals of the class are actually to teach students how we know how cells function. It's not so much about memorizing the list of facts as to how cells work. You can go and look that up. But rather, we're engaged in the process of how we know that.
What is the format of your course? Are there discussion sections, and is it taught via a lecture or seminar format, and are there clickers used in the course?
The course format is there are 2 sessions per week of an hour and 20 minutes each, and I would consider both of those discussion sections. So it's kind of a combination of a lecture and a discussion. The more of a discussion section it is every day, then the better the course went, in my opinion. The format is intentionally, I am attempting to reverse the order in which the science is taught. That instead of me telling students how cells work, I try to present an experiment and the result, and then we try to work backwards to understand how the cell works. That's much more reflective of how science and cell biology actually works. No scientist or cell biologist does cell biology by, there were no facts. You start with a blank sheet of paper and a question is like, how does this work? What can I do about it? And then you analyze the experiments. So every day we have a discussion about a particular piece of cell biology where we consider a problem, and we try to come up with the nature of the problem, and then we try to convert the problem into a hypothesis that could lead to an experiment. And then I know of a bunch of experiments that are already done historically, so I present the result, and then we usually break into smaller groups and discuss what that result means, and then come up with a new hypothesis, and that's the way every course goes if it went successfully. What was the other question? I'm sorry.
The last one is, are iClickers used in this course?
No, I do not use clickers. It seems to me that when clickers are used, sometimes students think that I'm spying on them in some way, like using that in the grade. So what I do is that at the beginning of the class, I hand out index cards, and then the students can fill those out with questions or comments, positive or negative, as long as they're constructive. And so that way they can ask me questions in something where it's totally anonymous. I can't memorize everybody's handwriting the way I care to, so you can turn those in, and it's a very comfortable way to ask me questions or make comments on the class and whether or not you think it's working. And so I read those. I then answer the mail at the beginning of the next class every time. So with a course of about 35 students this year, at the front end, I would get 20-some questions at the end of the lecture. Now it's sort of reached a steady state of about 5 questions or comments per lecture. So no to the clickers. This seems to be a way in which students feel more comfortable.
How are students in your course graded? Are there exams, quizzes, homework?
There are 4 take-home assignments. So the first 2 this time have been take-home exams, which are open note, open book, open library, open internet. Just do whatever you want, and then they're written. They're sort of short answer, like approximately 150-word answers, and there are 6 or 9 of those per exam. The only thing I ask is that students it's their own answers as opposed to working in groups. So those are the first 2 assignments. Then the second 2 assignments is more to assess the process of doing science. So this time I'm going to make an assignment where we pick a very controversial topic in cell biology. I'll pick them. And then I'll have students write a 3 to 5 page paper weighing the evidence of one side of that controversy versus the other side. I'll turn that in, I'll make comments on it, and I'll grade that. Then for the final exam, you'll turn in a revised version of that on the exact same question, and then I'll weight it. I'll make the final version of it like worth 2 to 3 times as many points as the first one. So the goal here is to be rewarding students for improving upon what they did.
Historically, which portion of the grade breakdown do students struggle with most?
I have no idea because this is the first time I've taught the course this way. So it's no longer a traditional lecture course. As I said before, it's completely reversed, where the information flow goes the other direction, where we come up with the experiment, we do the experiment, see the results, then figure out what it means. So it's an experiment right now. I'll be able to answer that next year.
Okay.
Yeah.
Are there points associated with attendance?
Not directly. I'm not sitting in there and taking attendance, but as you can well imagine, the way I described the way the course works, the only way to get anything out of it is to show up in class. So in that sense, yeah, I'm not scoring attendance, but you kind of have to be there in order to experience it.
Yeah. Are there required textbooks for the course?
No, I have suggested textbooks that are primarily for review, or sometimes for assignments. I mean, for the reading assignments for the class, it's usually primary literature. And then I list a series of questions that I think students should think about in advance before they come in. And usually I'll say something along the lines of like, you should have a Wikipedia or textbook level understanding of how proteins get into the endoplasmic reticulum, for example.
Do you provide students with PowerPoint slides or lecture outlines before class?
No, there are no PowerPoint slides. It's all the chalkboard, and this is all discussion. So there really are no slides. If I have a slide, I'll share it with the students, but usually there aren't any slides associated with the class.
What kind of background knowledge would a student need to know to take this course? Are there prerequisites?
I can't remember if there's a prerequisite for the class or not. I think that having the basic vocabulary of molecular cell biology helps. So if you've taken 250 and 252, that's more than sufficient to take the class. There are some students who are enrolled in the class who've never taken 250 or 252 because they're coming from different programs like in engineering and such. And all you have is just a little bit more catch-up time where you'll have to familiarize yourself with the vocabulary and the very, very basics of cell biology. So, yeah.
How do topics covered in your course apply to current research, medical, scientific advancements?
Well, I think they do. I think that we emphasize foundational experiments that sort of set the state, like our current understanding of how cells work. Then we try to transition that into some controversy or something that we don't understand, and there we explore more contemporary topics where there isn't agreement yet. And then how is it that you go about settling that controversy? Sometimes these have medical relevance, and other times they're just pure science.
What opportunities do you provide outside of class for students to ask questions or get assistance?
I have office hours, and you can schedule office hours, and then I'm also available after class as we're walking back. You can stop off. I usually have to grab coffee or a bite to eat, and there's another opportunity to meet with me. So plenty of opportunity for that.
That's great. Is there anything else you would like to tell us about your course?
Yes, I encourage you to take it and try it, especially if you're interested in going on and trying to become a professional scientist and going to graduate school or something like that, because it's an opportunity to see, at least I'm attempting to do this, to see how science is done. So if it's an advanced cell biology course, the point isn't to have you memorize more facts. That just doesn't make any sense to me. I think the point of the course is to see how is it that you do cell biology, to learn how cells work, and to sort of see the conserved elements in cell biology as how there's a framework of conserved mechanism where you can begin to understand the logic of cells, and also then begin to understand hopefully the logic of how to do experiments, the logic of how to think about creating a new hypothesis, and also the process of thinking about how to describe the significance of your work in a larger context, such as medical relevance, if you will, or a larger biology. So yes, I would encourage you to take it if you're interested in that.
Great. Thank you so much. Your course sounds really interesting. Thanks for meeting with me
Thanks so
today.
much, Lacey.